Just to mention to everyone that this webinar is being recorded. So again, thanks everyone. So my name is Jeroen Aerts. I'm based in Belgium, as I mentioned. I'm a customer technology advisor for Resolve Biosciences. And I actually have a background in neuroscience. I did a PhD in neuroscience. And afterwards, I did a postdoc in a pharmaceutical company. And before joining Resolve, I was actually working at VIB, which is a leading life science research institute in Belgium, and really at the forefront of single cell research. I was part of a team called TechWatch. We were basically scouting for early access technologies to get new technologies into the institute as early as possible across life sciences. I was specialized in single cell research. That's at one point more than, well, almost a year and a half ago, 2 years ago, I was doing a deep dive in the spatial transcriptomics and proteomics landscape, identifying all the major players and really understanding all the different chemistries and basically setting up collaborations with all the companies in the space. So I collaborated I collaborated with many companies, including Resolve Biosciences. So I used to be a customer, and that makes it, I think, very interesting for me to be now on the customer-facing side, of course. And so from VIB, knowing the competitive landscape, I decided to jump ship and choose for Resolve because I really believe that the technology that Resolve is developing is very, very powerful within the spatial transcriptomics field. And I hope to present some of the data to you to allow you to have a better understanding of what type of data that we can produce and what you can do with it, in fact. So in this Zoom webinar, we also have a little bit of an interactive idea. So I will be sharing some poll questions. It would be very nice to participate in that. I think it's very nice to learn from both sides where the interests lie in the spatial biology field. So without further ado, you can already see this nice intro slide where in the background you see a mouse brain section zoomed in on the hippocampus. And just to mention here that I think personally this looks like a piece of art, but every single dot here is an individual transcript. So that tells you already something about the resolution you can achieve with our technology. So, of course, as a way of introducing the importance of spatial biology, we know that in the recent years, we have made tremendous progress in understanding the cellular heterogeneity that lies within tissues. And this is mainly due to the massive adoption of technologies like single-cell RNA-seq, be it in droplets or in plates. But what you basically do is if you have your tissue, in this case, in this example, a mouse brain, you need to dissociate it completely. So you actually extract each individual cell, you encapsulate that in a droplet or in a plate from a well plate. And then you profile each transcriptome of each individual cell. And you will have some notion of where these cells reside, but it will be very crude. So the power of spatial biology in general is that we are now capable of really having that extra dimension where you analyze the same cells with their transcriptome in situ to really obtain a much clearer view of how these cells interact with each other, where they're located inside the tissue, and how they respond to, for example, drug treatment or in the case of pathology. So I want to already launch, let's say, the first poll question here to better understand our audience. And the first question is, who of you is already currently using spatial biology tools? So yes, no, or you dialed into the webinar today wanting to really get your hands on some spatial biology tools and want to use it within 6 months, or are you still doubting and want to start off maybe in the coming years? So please feel free to select one of your choices. and I will share the panel results in a minute. I cannot see the results. Yes, now I can see it. Okay, that's actually quite nice. So we have already 35% of the people that have experience with spatial biology tools. 44% does not have experience, but it's nice to see that 12% wants to use it within 6 months. And a minority, let's say 9% in this case, wants to start early next year or starting next year. So it's nice to see that there's already many people here dialed in that have experience with spatial biology tools. So it would be very interesting to know also during the presentation what kind of questions you have and how you feel about the technology we offer. So going ahead, so I want to show you a bit more on the technology we're developing at Resolve Biosciences. So the platform we're developing is called molecular cartography. And I want to show you an example on the mouse brain. So typically, of course, a mouse brain, I consider it as a benchmarking tissue for spatial biology tools. So this is really a tissue type that is very well understood and very suited to really demonstrate the power of the technology. So we applied our technology to a part of the cross-section through one of the hemispheres of the mouse brain, as depicted with the mouse brain atlas here. And on top here, you can see a projection of the data that we captured for 100 genes, and we were able to identify or quantify over 10 million individual molecules. So this tells you something about the data density that we capture and also a bit about the resolution already. So, but what is at the heart of our technology? What's at the core? So how does it work? We have an image-based technology based on combinatorial single molecule FISH. So basically, if you have your tissue section, what we do is if you have, let's say, your 100 genes that you want to target, so it's a targeted methodology, we design transcript-specific probes, and if you have your RNA inside your tissue, basically we hybridize tens of different probes that cover the transcript, and we have a proprietary chemistry that allows you to colorize, but also decolorize these transcript-specific probes, and we use that colorization and decolorization chemistry to obtain through cyclical imaging methodologies, really a sort of barcode for every transcripts. And that's illustrated on the right side. So let's say we know by the way we design our probes that for transcript A, so all the transcripts originating from target gene A, we know that in first round of imaging, it will light up in one of our channels, let's say the green channel. And as you can see here in the cartoon figure of neuron here, we can pinpoint exactly the location. And we know in the second round for the same transcript, we expect it again in the green channel. For the round 3, we expect it in another channel, the red channel here, et cetera, et cetera. So after X amount of imaging rounds, you can actually see already that we have a unique barcode for each transcript. And we can actually decode at the very end of all these imaging cycles, we can decode the identity of that individual transcript in a spatial dimension. So we know in this spot where we see all these fluorescent dots appearing at the very end, we know this is transcript species A. And this gives a very nice basis for specificity, also for sensitivity, because this is based on single molecule FISH. And it also allows for digital quantification because this is not based on fluorescent intensity. We just at the very end decode the identity. And this is giving you with XYZ coordinates a digital quantification method. This is further illustrated with the slide I show here. So more on the specificity, of course, it's built into the chemistry, but we validated it with just comparisons with traditional in situ hybridization. So these are different cortical layer markers for mouse brain cortex. And we depicted here in the same data set, this individual genes in green. And as you can see, it nicely recapitulates what is known from literature. Two big differences again. So with traditional in situ hybridization, you can only target, of course, one gene. Here in each panel, we just colored each gene green for the sense of interpretation, but each panel actually contains information from 100 genes in a single section. And again, on the digital quantification, so here we don't use any optical density or fluorescent intensity. We can really do transcript counting, because each spot we identify the transcript identity. And then more on sensitivity. I think sensitivity is very important in spatial biology. There are many other technologies that do capture a lot of spatial information, but not necessarily also the very low expressed genes. And I think when we talk about sensitivity, of course, we can show you correlations with bulk RNA-seq, even though these data sets are from different sources. We very often see very nice correlations. This is really as a sanity check to see that our data sets make sense, of course. But I think the comparison that is most important is really comparing that to the gold standard of sensitive spatial technologies, and that is single molecule FISH. And when we check our platform on a gene-to-gene basis, we see the same amount of counts per cell that we would expect from our technology based on the chemistry that we developed this on. Is the single molecule FISH-based chemistry. So it's nice to see that also the sensitivity is very high. So we do capture almost basically all the transcripts within a given tissue based on what we know from single molecule FISH. So going further into the data set on the mouse brain, I will show you also other data sets just to show you what you can do with it. So if you show, if we just show here some cortical layer markers, just 13 out of the 100 genes, you can already very nicely see that we are able to recapitulate what is known from this tissue, where we just highlight in different colors in a nice rainbow figure, indeed, the different layers within the cortex. But you can go further into that and really show, for example, if you just add on to that more cell type markers, so 35 of the 100 genes, you can clearly see that we're able to distinguish glutamatergic neurons in red. We are able to distinguish these yellow cells here, which are the GABAergic interneurons. Also, some microglia, astrocytes, oligodendrocytes are nicely interspersed in the sea of red, which are the glutamatergic neurons. But of course, this is very dense data that you visualize here. Our visual system is not able to really make any or a lot of sense out of this data. So what is nice, of course, you can just really zoom in on, let's say, the more cellular level, individual cells. You can visualize, for example, the different subtypes of inhibitory neurons. And in this case, I show you here some markers for the different types of inhibitory neurons, like GAT1, parvalbumin, somatostatin, VIP. And this already nicely shows you, when you zoom in on the mouse cortex here, that each individual cell indeed has these transcripts expressed. And for example, here is a very nice parvalbumin positive interneuron. You can even see these axons, dendrites popping out of the cell body, which I think is already very nice to see. And this is a demonstration basically of the resolution and also the sensitivity, I would say, from our technology. Is the resolution we have is really diffraction limited. It's single molecule. And here is a demonstration of all the individual somatostatin transcripts located in this one cell. And also to illustrate here that from these 10 micrometer sections that we apply our technology on, we also capture all the 3D information. So we take Z stacks throughout the 10 micrometer tissue section. And this allows also for increased specificity for spot calling, but also allows you to really dive deep on a subcellular resolution, really on a single molecule resolution. For example, to show or analyze differences in location of transcripts within a given cell based on what's happening in the neighboring cells, or based on treatment or pathology. So going further in other types of analysis that you can do with a data set like this, you can actually segment out each cell and get some pseudo single cell data out of this. And here we applied a very robust single cell segmentation algorithm from QPOT. So this is based on DAPI. So you take your DAPI nucleus, you expand the selection, and you consider that as a single cell. Of course, this is not 100% accurate, but it's a very robust automated way of doing this. And this already gives you an idea of the amount of signals that you capture in a given cell, or the amount of genes per cell that are actually expressed. But of course, this differs a lot with how you tailor the gene list that you apply to your tissue section. So it allows you to really extract the cell to gene or gene to cell matrix, and you can import that into tools like Seurat to generate UMAP clusters. So this is very reminiscent of what we're used to when dealing with single cell RNA-seq data. So here we clustered around 35,000 individual cells. And here you can see that we are able to identify all the major cell type clusters. And also in these heat maps, you're able to really distinguish all the different marker genes that make up the identity of these clusters. And I think what is very powerful with our technology, but mainly also with spatial biology in general, you can take these clusters and actually put them back onto the tissue. So this allows you to really map all these different cell clusters and look into changes. How does the distribution change based on pathology or a drug treatment, for example. And this is also a slide illustrating the same thing. So it really allows you to just map all these individual clusters back onto the tissue section and do simple analysis, for example, like counting how many astrocytes are there in this particular tissue section. And this actually nicely recapitulates what is known in literature to the dot almost. So that's also another validation of, even with a robust cell segmentation algorithm like I showed you with QPOT, we're able to gain a lot of insight and really have a lot of robust data coming out from that. So further on, I want to illustrate some more use cases still within neuroscience, but this is a use case where we focus on disease-associated microglia. So these are a subtype of microglia, immune cells in the brain that really are very tightly associated with the amyloid plaques that are so reminiscent or so known, let's say, within the Alzheimer's disease models. So what we did together with a pharmaceutical company is really comparing wild type mice with an Alzheimer's mouse model. And here we labeled again with these markers for neurons, astrocytes, microglia, etc. So part of the cortex, you can actually see big differences already, of course, a loss of neurons and also an increase in these microglia here in green. And they surround or seem to surround the amyloid plaques here. And just for your information, so we labeled the tissue for these amyloid plaques with a specific dye that binds to these amyloids. So our technology is designed in such a way that the tissue remains intact throughout the transcriptomic assay. So you can actually do in situ immunohistochemistry, so antibody labeling or dye labeling after you performed our assay. So I think that's a great feature. Also, if you're interested in really obtaining some multi-omic data sets with other tools you might have, you can take the tissue and really perform any assay you want afterwards. So we don't remove any protein information during the run. So other things you can do is, for example, map which genes are enriched in which condition of your cortex. So here, for example, here in green in the Alzheimer mouse model, we see that all the enriched genes in our data set are actually very tightly located surrounding the amyloid plaque. So the biggest differences in this data set are really very spatially segregated. So again, you can do cell clustering as I showed you before. So identify all the different cell types here and really correlate that to pathology. This is what has already been done previously just based on single-cell RNA-seq. So you can really map the diseased state with the wild-type state. And as you can see, we again can identify these disease-associated microglia, so as a separate cluster within the microglia. But I think what is very nice here as a case study is that you can actually take the spatial dimension here and really see that if you quantify the distance to the center of the plaque, you can actually very nicely see that for these microglia, the closer you get to the plaque, the more you're into these disease-associated microglial clusters that you can see here. And this is illustrating a similar thing. So if you really analyze the amount of cells, for example, between the both conditions, you of course see that as illustrated before, you have a loss in glutamatergic neurons and an increase in these microglia, mainly surrounding the plaque, and that's actually quantified here. So we quantified actually the distance to the plaque, and the more you go to the center, you see again this drop in glutamatergic neurons and this increase in microglia. So this gives more insight into what we've known previously from single-cell RNA-seq, that you have this spatial dimension really pinning it down to the involvement of these amyloid plaques within the brain. And you can do that also on a single molecule, a single transcript level. So we analyzed over 600 individual plaques. We drew concentric circles around the plaque, and we just measured the expression, or we counted actually the individual expression of these disease-associated microglia, these DAM markers. And as you can see here again, the distance to the center of the plaque, actually, when you go closer, you see an increase of the counts of these DAM markers, and also you can see a decrease of neuron markers. So very similar, but then really on a single molecule level. You can also zoom out a little bit and really quantify and really illustrate also on a cell cluster level or cell identity level what happens differently in these mice. So on the right side, you have these wild types. You can visualize homeostatic microglia, astrocyte reactive astrocytes that are bordering, let's say, this cortex here. And if you compare that to our Alzheimer mouse model, you can clearly see that a lot of things change. You see these dams popping up. You really see these reactive astrocytes dispersed throughout the cortex. So many different things are ongoing, and I think the spatial dimension really allows you to have much more insight into this. We also tried this, or we're also studying this actually on human clinical post-mortem brains, control brains, but also BRAC6 stage Alzheimer human brains, just to show you that we can also process human samples. I can show you other examples of that as well. And it works on human brain, and you can clearly see already big differences in just the amount of neurons present here and how these microglia seem to pop up as well. And this is an ongoing study, but very interesting that we can translate also that those findings in a spatial context in the human brain. So going beyond neuroscience, I want to touch upon what we've been doing on liver. And liver is a very interesting tissue from a spatial perspective, because you really have this very nice interplay between central veins and portal veins, and how these hepatocytes, so these liver cells, are actually interspersed between these specialized veins. And of course, you also have these cholangiocytes that produce bile within the bile duct. So a lot of things are happening in the liver tissue, which with very interesting spatial dimensions here. And I want to show you a study we did in the context of, well, with a customer actually, with 100 genes, and just to show you some high reproducibility within a run. So these are actually biological replicates. So from different mice, we took a slice of a liver sample and we correlated all those individual counts. So you see a very high correlation of all these signals, and also a very high correlation with public liver RNA-seq data set. So this is also a nice thing to know, that we can obtain a high reproducibility even between biological replicates from the same control tissue in this case. So this also helps us understand how we should approach experimental setups when it comes to highly multiplexed spatial biology tools. So more on this liver data set. So this is just an image of 10 representative genes for different markers. And as you can clearly see, so we see, for example, surrounding this central vein here centrally in the image, that you have really a gradient of a specific gene, CYP2E1 here, and also some marker genes here for endothelial cells that line the central vein, and some specific genes here that are really having a certain expression pattern going further away from the central vein. It really changes a lot in how they express these genes. And this is, again, zoomed in on the central vein. So CYP2E1 and GLUL are really markers for different hepatocytes. And we validated that, or this was also part of validation, what we know from literature already. In this earlier study, you can see that for GLUL, we indeed see, let's say, a single cell layer surrounding the central vein, while CYP2E1 more has a gradient. And this already shows you, again, a big difference between traditional single molecule FISH and the type of data and resolution we can obtain with our data set. So it's more quantifiable, I would say, and we can nicely recapitulate GLUL here is indeed expressed in just a single cell layer, with the CYP2E1 really having a more gradient-like expression. And the same goes for, again, the same data set where we visualize these hepatocytes with just in a sea of red. And then you have Kupffer cells in between. You have some hematopoietic lineage cells. You have cholangiocytes. So you can clearly see that even in this sea of red, where you can think, wow, where is my rare cell type that I want to visualize, you can actually see these Kupffer cells, for example, nicely popping up with the marker genes we selected. And also in the central vein, you can see here in purple, these endothelial cells, again, really nicely aligning the central vein here. And also even these cholangiocytes that produce the bile for the bile duct, you can actually nicely see what we expect, the bile ducts really being in close proximity to the portal vein. You can actually see here in green that these cholangiocytes do express the proper marker genes here. And what we did, or what you can do again with this data set, is really do again a cell segmentation, do the clustering, and you're able to then identify many different subtypes of hepatocytes. And only looking at these hepatocytes and mapping those different clusters back on the tissue section, I think it's very beautiful to see that you have a very nice gradient of different subtypes of hepatocytes depending on where they're located in spatial relation to the central vein as opposed to the portal vein here. So that's already, again, a nice application for really studying changes in these subtypes of hepatocytes upon, for example, inflammation, or when you have a very fatty liver, for example. How do these hepatocytes change spatially? And just to comment a bit further, so I mentioned already that our technology is compatible with immunohistochemistry, but we've also done some studies with customers that already have a lot of immunohistochemistry data, just to show that even between different samples, there is a very high correlation in just what we expect from transcripts where proteins and mRNAs are located. And this is, again, summarizing where GLUL is located around the central vein here in red. So it's really recapitulating the same expression pattern. Same goes for the light blue here in EpCAM, here in blue again, also EpCAM surrounding the portal vein. So the correlation is definitely there, and this also goes much deeper. Let's say if we look at the stellate cells and the Kupffer cells nicely, tightly packed together, we see actually very similar mRNA counts for these different cell types, also very closely packed together. So it's nice to see that even between different sections and between historic data sets from customer, you could actually retrieve nice data from our molecular cartography platform. So I showed you some brain, I showed you some liver. I think at Resolve, we are very much aware that within spatial biology, there are many spatial questions out there for many different tissue types. And what I think is great about Resolve is that we really try to tackle as many different sample types as we can think of. And it's great to see that we already generated data on many different species, many sample types. As you can see here on the left, this is actually a data set on plant tissue. This is maize root. In the center, you can see a reptilian brain. And on the right side, top right side here is a cross section through the zebrafish head, where you can actually clearly see here in green also the retina. And it's really a nice feature, I think, to be able to apply all these exciting, highly multiplexed chemistries from our platform onto different tissue types. So I want to raise another question for you, knowing that, okay, we explore a lot of different fields within spatial biology. I want to know from you, the audience, which fields do you see as most important now at this point in spatial biology, and where indeed where do you see it apply to? Do you think neuroscience is the most important, oncology, or maybe immunology, diagnostics, or even drug development? And I understand this probably has to do with your individual background, but it would be nice to see also what type of backgrounds we have within the audience, or where you actually see spatial biology being the most important currently. So we're still waiting for some answers to pop up. Yeah. Okay, great. So we have some results in. So the majority of people here, they consider oncology as the most important field to have spatial biology apply to. I think that's a great insight as well. I mean, there's a lot of important questions to be answered. For example, in the tumor microenvironment, I see 19% of the people ticked off immunology. I think immunology and oncology, of course, are really also sometimes very intertwined, of course, when you're dealing with the immune cells infiltrating in the tumor microenvironment. So both fields, I think, are very important. And of course, neuroscience, I already showed you quite a lot of data on that. So 29% of the people think neuroscience is really a major field where spatial biology is really important. And I couldn't agree more. I mean, neuroscience, next to oncology, I think, is the field where we see a lot of applications. One of the applications I showed you, of course, is Alzheimer's disease, but you can think of many others. And then 10% here voted for drug development. I think indeed, when we see at what pharmaceutical companies are doing, how they apply spatial biology for their drug development efforts, it's very interesting. And then only 2% of you highlighted diagnostics. And actually, this is very reminiscent of or very typical of where the field is at the moment. We also do realize that diagnostics is more further in the future, and we need to tackle some challenges in really adopting or getting the technology adopted by the diagnostics field. Okay, so thanks a lot, everyone, for including those answers. It's very insightful. So moving on, so I mentioned already the tissue plexity. So we tested our technology on many, many different sample types, plant, reptilian brain, very exotic things. But of course, we also tested our technology on samples within oncology, and it's nice to see that many of you voted for oncology. I just want to show you here a snippet of a data set where we applied our technology on breast cancer, so human breast cancer, just highlighting just 14 genes in this data set. So going through these genes, you can already see that many of these genes are really expressing a lot of transcripts. The data is really dense, even when packing just 14 genes. And what is so striking, I think, is that you see a big difference between the two patients. So this is also a nice way, I think, or a nice case study of really mapping patient diversity and where that diversity is located. And on top, of course, you can really zoom in on the single cell level, quantifying transcriptional changes that are happening in the tumor microenvironment, really zooming in on these tissue types. So going further in the different applications, so this is an example of heart tissue where I highlighted just some markers for myocardiocytes, smooth muscle cells, just to show you that also in cardiology, there are many spatial questions that need to be answered. And this is a mouse model for cardiac disease, I think. And this is just showing you some basic, this I think is a wild type example actually. This is showing you the nice spatial distribution of these smooth muscle cells and these fibrotic markers and things like that. So that's very nice to see that also in cardiology, you see very nice spatially distributed transcripts, of course. And I think the timing is upon us, of course, to also do some SARS-CoV-2 related work. We have done so, and we are currently doing a lot of that in many different tissue types. But I want to show you here an example also from a cell culture assay. So we can apply our chemistry to cell cultures. And this is an example where we probed many different genes, but also the viral transcripts here are highlighted in yellow. So we just increased the size of the dots here of the viral transcript just for easier interpretation. So the it's not that the resolution is different, of course, but with our visualization tools, you can actually change the diameter of every point to really make it much more suitable for interpretation. So compatible with cell cultures, and also if you're interested in SARS-CoV-2 related work. So I think it also has many nice applications with spatial biology. So just more of a summary. So with our technology, we can do a lot of different species, a lot of different tissue types, also many different cell cultures, and also organoids and things like that. We offer up to 100 genes simultaneously in a given sample. I think our resolution is really great for many applications. It's really subcellular, single molecule, and we do capture the full 3D information from a single 10 micrometer section. As far as bioinformatics goes, we do have an interactive software tool where you can easily visualize. It's also compatible with cell segmentation tools with Seurat for clustering. We also have a tool which is more of an ImageJ plugin. So we do understand that many people already have a lot of tools available that they use in their lab for image processing. So we wanted to make it as open as possible. So this ImageJ plugin really allows you to use the whole power of ImageJ and really import and export basically whatever data format that you want. And for example, import cell segmentation ROIs from QPath or any other tool, export that's cell to gene matrix and import that back into your Seurat pipeline to really do that cell clustering, etc., etc. So it's very open. And we also provide you with some R packages to really insert that into a more elaborate data analysis pipeline that you may have. True power in our technology is also the digital quantification. So one spot is one transcript, so you can really go into transcript counting. Our technology allows you to have the sample completely intact after the assay. And that's really important to have the ability to combine it with immunohistochemistry or any dye labeling or maybe other types of analysis that you want to do. So we do not clear the sample. We do not remove the proteins during our run. We are developing or planning to set out instruments in the field that actually can accommodate 24 samples in a single run. So we do provide glass slides with 8 different wells, and you can place a tissue in 8 of those individual wells. And the instrument can hold 3 of those glass slides. So for a total of 24 samples, you can analyze in a single run for those 100 genes per sample. So the sensitivity is really, I think, a true strength of our technology. We don't use any enzymes, or we don't have, let's say, a type of capture technology. For example, if you compare that in terms of sensitivity and resolution, we see that For example, these sequencing-based methodologies where you permeabilize your tissue, you capture your RNA, you put it on a sequencer. Very often the resolution is quite low there, but also the capture sensitivity. So you mainly capture highly expressed or mid-expressed genes. If we compare our technology to other technologies that use, for example, padlock probes, where you need to amplify the signal through rolling circle amplification, or any other technology that uses a lot of amplification, this is actually not the most sensitive strategy. Our strategy is really to use the single molecule fish chemistry to gain the highest sensitivity. And again, listed here on the slide is the tissue integrity for analysis. That is very important. So I want to have another poll question here. Just having shown you everything here in terms of parameters that we offer in terms of multiplexing, also the sensitivity, the resolution, field of view, which is a topic we can also discuss, and also the throughput. So what do you think for your application, for your research, what is the single most important parameter or capability of a technology that you're looking for? And I think this is a very important question for many technologies out there, but also for us to know where our strengths and maybe potentially our weaknesses lie. But also we want to make sure that we can help you deliver the science that you need, of course. So I'm very curious for this one, what people will answer. So I'm waiting for the results. Okay. Great. Wow. It's a bit of a mix, I would say. So I think 26% of you voted for multiplexing. So almost an equal amount of people, 24% voted for resolution. Then the third in line is sensitivity, and then throughput. And then as the last aspect is a field of view. So that's actually quite interesting to see that it's really a mix, and maybe if we would add or would have added the last choices, all of the above, I think many of you probably would select that. So I think a lot of these parameters are very important, and I think it's a testament just that the capabilities of a platform are really tightly bound to, of course, the applications where you can use it, or what type of research you're doing and what kind of questions you have, that will map beautifully with our technology, or maybe other aspects might be less suitable. So I think this is really also nicely reflected in the questions, but it's nice to see that resolution and sensitivity, of course, which I think our technology is really excelling in, is very important. And of course, the multiplexing 100 genes, I think typically is already quite a lot for many types of research. So that's nice to see. So thanks a lot, everyone, for providing your answers here. Okay, so let's see. I just want to also end with, so I talked a little bit about, okay, we're planning to deploy some instruments in the field, but of course, we are also offering from today, not today, but already for a while, we are offering currently an end-to-end service and support. So what that entails is, if you have your favorite gene list, we can design the probes, we order the probes, we ship you the glass slides. So these are just plain glass slides. They're coded for just better tissue adherence, but they're not in any way activated or anything. So if you want to have multiple tries of a tissue section, or if it folds, it doesn't really matter. You just take additional sections, you fill up the slide, and you can you only select the best sections for us to proceed with the analysis. So you prepare those slides, you ship them to us, and we do everything else. We just run the entire experiment. We do the primary analysis, we do some secondary analysis, and we set up a meeting with you to really go over all the details. We provide you with a full data report and provide you with all the tools, as I mentioned, the visualization tool, the image shape plugin, some R packages if you want, and we really also assists you in really analyzing your data and interpreting your data according to your biological question. So I think for us, it's really important that you as a customer can use our data and really make the new discoveries that you want. So I actually want to end our Zoom or webinar with a trivia question. So I hope everyone plays along with this one. So there's one trivia question we want to ask you, and you can already see it here. So our Resolve logo here with the RE, it's very reminiscent of one of the elements in the table of elements here, and called rhenium. And rhenium is like a silver grayish metal, and was discovered in 1925 by 3 German chemists. And the question here I have is the following: where was rhenium discovered? And the first one that has the correct answer, you can put it in the chat. They will win a prize. So I'm curious to see who has the correct answer the first. And it also has a more personalized meaning for Resolve Biosciences. I can imagine everyone Googling now. Okay, I see actually one answer. And it's already the correct one by Michel Molenberg, if I pronounce that correctly. So the correct answer is it's discovered near the Rhine. And that's actually a bit of a history of our company. We are located in Monheim. So Monheim is very close to the Rhine. This is a picture actually of the campus we're located in. And in the background, you can see actually the Rhine River, and it's a beautiful surrounding also to walk around, to have a run. It's really nice to have such a nice location in Germany. So yeah, so thanks a lot. We will follow up with Michel for that prize. And it was very nice to see so much interaction with this polling. I hope you enjoyed the webinar here, and I see also a lot of questions popping up, so I think we still have time, of course, to go over the questions. Let me see. Okay, so I see a first question here popping up from Benson Lee. So the question is single molecule FISH different from in situ sequencing? That's actually a good question, just to elaborate more on the different spatial technologies you have. So single molecule FISH is different from in situ sequencing. So in situ sequencing, how we, let's say, define it is, these are the technologies, for example, like Cartana or ReadCore developed. So these are the FISH-seq or in situ sequencing technologies that use padlock probes that can target a specific part of the sequence. So these are very powerful technologies, for example, to do or target specific SNPs, for example. But the downside there, I think, is you are actually performing some enzymatic reactions in your tissue, because in situ sequencing, you actually need to amplify your signal through rolling circle amplification. So you have your padlock probe, you do that rolling circle amplification, and you generate these little nanoballs inside your tissue. And what happens is, if you then decode it with your target probes with fluorescent tag on top of it, you actually have a fluorescent dot that is quite large. So this is typically around 600 nanometers. So comparing that to the dot size we have, around 250 nanometers, that's quite big. And also these enzymatic reactions are not 100% efficient, of course. No enzymatic reaction is. So that actually eats away a little bit at the sensitivity. So typically with in situ sequencing, you see lower sensitivity in the amount of transcripts that you capture. While in single molecule FISH, you have your RNA, as I showed you, and you cover it with target probes, and then you detect those target probes through different chemistries. So that's actually a big difference. So in single molecule FISH, you don't use any of those amplification methods. So I hope that answers your question, Benson. So Michel, Michelle Molenberg asked, what is the maximum size of tissue that can be analyzed at once? You show pictures of a whole brain. Actually, that's a good question. So with our technology, you can actually image a whole brain. Basically, it's actually a hemisphere, so it's still not the whole brain, but the whole hemisphere. But in terms of throughput, there's always, of course, when dealing with an imaging-based technology, always a trade-off. There is a trade-off between the amount of genes that you want to target, the resolution that you want to have, and the field of view, so the size of the image that you want to capture as an image. So we can do that, but if you look at the throughput, what we typically do, and this is really what we offer in our service, for example, but also in the instrument. In the instrument, you have more flexibility, but in the service, for example, what we do is we try to have more samples that you can analyze. We provide you with bright field images, and you can actually do a region of interest selection on those bright field images to really zoom in only on the region of interest that you want to analyze for your specific question. And typically then it ranges per tissue section, let's say several square millimeters that we zoom into, but this actually increases your throughput. And the way we designed our instrument is where you can place all these different glass slides. It allows you to actually better compare different, let's say, patient samples or wild type knockout or any other treatment condition by placing each condition separately in a well. Instead of having, let's say, one big surface where you need to put in several tissue sections, for example, and have a big field of view, but it's very difficult to identify which sample is which one. So I hope that answers your question, Michelle. Then an anonymous attendee, can you stack together several tissue sections to 3D reconstruct the tissue? That's a very interesting question. I think it's a very nice application of spatial biology. So of course, for now, we have focused a lot on the X-Y axis, but the Z-axis is equally important. And we see many people wanting to really zoom in also on that Z-axis. So I mentioned already that we do capture everything in the Z-axis, but only in the 10 micrometer section. So we are actually actively developing tools to actually reconstruct that 8 tissue sections or even the 24 tissue sections after a full run bioinformatically in a 3D model. So for example, if you have your 8 sections, what we hopefully then soon can offer, if we develop this, is really offer you also, let's say, the option to go for an 80 micrometer depth 3D model, or even stack that up with additional slides and reconstruct it throughout a larger Z-axis. So that's definitely on the planning and super important, I think. Another question from Michelle. So is 100 the maximum number of genes analyzed simultaneously? So for now, this is what we do offer. So this is currently the maximum, but it is on the roadmap to actually increase this. So we can definitely increase this. I mean, as I showed you, we have, let's say, a barcoding mechanism. We use cyclical imaging rounds. And that allows you to, for example, add another channel or really extend your barcode and things like that. So it's definitely possible, and we're working to increase this number while also maintaining, let's say, our throughput. And because when you want to image more, this has an effect, of course, on your throughput and also on your field of view. But this is actively being developed. Let me see. I see some other questions popping up. Okay, so another question from Benson. Can the technology be used with high-plexed immunohistochemistry, such as Polaris, Codex, or Maxima? I would say yes, that's the short answer. The longer answer is, of course, that still needs to be tested. I think this is a very interesting use case of generating highly multiplexed multi-omic data. So as I mentioned, our tissue remains intact throughout the entire assay. Of course, things to keep in mind, because we have, well, we do treat the tissue. Well, the tissue undergoes several steps with hybridization and some other buffers, etc. It could be that for some antibodies, the availability of some antigens might have changed a little bit. So this probably needs some optimization. And for just low-plexed immunohistochemistry, this is quite straightforward. But with Maxima and other technologies like Akoya Biosciences or other technologies, this I think is definitely possible, because the proteins are still in there, but it might need some more optimization, of course, because inherently these technologies are already quite tedious to optimize, because you need to optimize the titration of all these antibodies anyhow. So, but theoretically, definitely possible, and I'm super interested also to see some data on this. And internally in the company, we're also actively exploring, let's say, building multi-omic data sets like that. Another question from Benson: can this technique observe SNPs? As I mentioned before, so that's one of the differentiators between, let's say, the in situ sequencing technology and our technology. Inherently, with single molecule FISH, it's not possible to detect the SNPs. So SNPs indeed are, let's say, too short, or yeah, it's just one nucleotide, of course, to really allow for the differentiation between the specificity of probe design. So we need to cover a certain part of the RNA to really have that specificity built in. So SNPs is unfortunately not an option. What I can say maybe related to that, the minimum length we need of an RNA to be able to design specific probes against that is 500 nucleotides. And 500 nucleotides is around twofold shorter than other technologies like MERFISH, for example. They typically need a longer sequence to actually be able to design specific probes and have that specificity. And that allows for our technology, I think, for a lot more of the genome that you can interrogate. And we have internal lists of genes that are related to disease that are indeed targetable by our technology, that are in between, let's say, the 500 and the 1,000 nucleotides in length. So I also think that's quite interesting within the single molecule FISH space, that are still very much differentiators there. Lemuel Racacho, if I pronounce that name correctly, has a very interesting question. So is there any work on creating standards or best practices for spatial transcriptomics, especially in clinical labs that would like to adopt this? That's a very important question. I think actively in the spatial community, these questions are being asked and try to be answered, but I think the field in general is a bit too young. I think we need to work together as a community to really establish these standards and really more widely adopt these standards. I think it goes from the beginning all the way to the end. Let's say in the beginning is tissue preparation, how to really standardize the correct way to prepare all the samples and really have the least variability in how to prepare these samples for a specific technology, and more ideally for many types of technologies. On the other hand, of course, is really the data analysis, really standardizing many of the open questions in how to deal with, I don't know, quality metrics and how to really assess and standardize data analysis in general. Those are still open questions. I think we at Resolve are also actively tackling many of those questions. And I think broader in the spatial community, I think everyone is aware of these questions, and I think it's a matter of time. But I see spatial biology, spatial technologies really being adopted by the clinic in the coming years. Let's say, especially within 5 to 10 years, that would be ideal. But many challenges need to be overcome still. Okay, I don't see any open questions anymore. Maybe the chat. I see another one from Frederik de Smet, who I know from K Leuven, actually. So nice nice of you to join, Frederik. So how does your technology perform on old FFPE sections? So that's a good question. So we have done FFPE work. So our technology works, let's say, with FFPE, with paraffin embedding. But of course, not all FFPE is created equal. You have, of course, the effect of RNA integrity. And your question really holds to old FFPE sections. Of course, it really depends on how old, how long were they stored, how were they prepared, etc. And looking into the RNA integrity, of course, if your RIN values, for example, are very low, I think, yeah, we cannot really promise that our technology would work. I don't think any other technology would work very well when you consider capturing enough data points. But I think that's still ongoing in really assessing how can we correlate, let's say, the quality of the RNA to be able to assess what is the success rate of our technology on these types of samples. But chemically speaking, from an FFPE embedding type of thing, it will definitely work, I think. So I think it's also important generally to really push for that. It's on our roadmap for sure to really assess what type of samples we can process. What I think as well is very important, we are talking about RNA quality. And we are used to that in terms of RIN value, for example. This is more of a bulk thing that you apply, and you take a piece of the tissue, you determine the RNA quality, but this can also be different in a spatial dimension, of course. Some parts of the maybe old FFPE sections will still have very nice RNA, while other parts of the tissue section will have maybe very degraded RNA. And I think we should aim to develop these spatial RIN value type of, or spatial RNA integrity tools to really have a better QC upfront before we decide to run our assay. So I think these are all very important questions and tools that still need to be developed, but FFPE is on the radar, and we do have already some experience. That's what I definitely want to mention. So thanks for that question. Okay, I don't see any other questions popping up, and I also see we are already a bit over time. So I do want to thank everyone for attending this webinar. It was a pleasure seeing so much response, so many questions, and also the interactions with the poll questions. So thank you very much. If you would have any other questions, feel free to reach out to anyone within Resolve. We're happy to take on your questions. And also, if you're interested in running projects with us and running samples, we are definitely able or willing to help out and see how we can benefit your research. So thanks a lot. I will stop sharing my screen for now, and I think we can end the meeting here. Thank you very much. Have a nice evening or a nice day.